Thank you, Tessa, and thank you everyone for joining us today. We are very happy to have Dr. Summerfield today for our webinar series, and this will be the last webinar series of the year. So we will start over next year in February. So I'm going to introduce Arthur, who is a veterinary immunologist with particular interest on immune response to infectious diseases and vaccines in pig and ruminants. His research is focused on antigen presenting cells, viral immunology, as well as understanding and developing novel immunotherapeutics and vaccines using systems immunology approach. If you read Dr. Summerfield's paper, he's very creative in the way he approaches the different questions that we have regarding the immune response. He has a very long trajectory, and after visiting school in the US, France, and Germany, he studied veterinary medicine in Berlin and obtained his PhD in 1994 in Tübingen at the Federal Research Center for Viral Diseases, now the FLI, the Friedrich Loeffler Institute in Germany. Then he moved to the Institute of Virology and Immunology in Mittelhausen. I don't know if I pronounced that correctly, in Switzerland, more or less. In 2006, he became head of the Laboratory of Immunology, and in 2010, head of the research development of the IVI. In 2013, he was nominated professor for veterinary immunology at the veterinary faculty of the University of Bern. And he has published a lot of papers, very exciting papers as well. And this one in particular, the one he's presenting, the work he's presenting us today, is part of our research topic that we publish as the GFRA community in Frontiers in Veterinary Science. So Arthur, it's an enormous pleasure and an honor to have you today. We are really excited about your presentation. So the floor is yours, and thank you so much for joining us today.
Thank you very much, Alejandra. So I need to reshare the screen now. Good. So this work actually was performed about 10 years ago and was sitting in my drawer for 10 years. The people that did the work actually, some of them retired. So Heidi Gerber was that, and Emiliana Brocchi. Some of you might know her. She also retired, but I was able to take some of the time and say, okay, I should publish that. Because I think it has interesting aspects for the field of foot and mouth disease immunology. So it's about antibodies and FMDV. And for those who are not so familiar with the field, just as an introduction, we know that neutralizing antibodies are quite a good correlative protection, but not absolute. You have to know that there's no cross-reactivity between the serotype, and that also within serotype, there is also such a big heterogeneity that you cannot assume cross-reactivity. So what's required for neutralization is really high affinity, or alternatively very high antibody concentrations to get neutralization. I'm talking about in vitro. And the second point is clearly, like for many RNA viruses, they are a main driver of antigenic escape and virus evolution of foot and mouth disease virus. So what's happening in the field is, of course, you lose immunity to field viruses or vaccine strains over time, and you end up with a very high antigenic diversity of field strain in endemic situations. The third point is that we now know for quite some time that non-neutralizing antibodies also mediate antiviral functions through opsonization. So basically you have immune complexes that bind to Fc receptors that we have on antigen-presenting cells, on macrophages, neutrophils, but also on natural killer cells. And in the phagocytes, that results in uptake of the immune complexes and, for instance, destruction. And what we know from in vivo studies is that this process is actually essential because you need to remove those immune complexes for a successful immune response. And the question why is very simple. So the basics of an antibody-antigen interaction is that this is a non-covalent binding, so it's reversible in nature, and we'll follow the formula that you see on that screen, where you have the K is the affinity constant that will be defined by the amount of antigen antibody that is bound together divided by free antigen and free virus, or free antibody and antigen. And so this is impacted by many factors that are also present in the body, like temperature, pH, ionic strength. And so that might be different in different tissue and enable, if you have a weak affinity, so a low Ka might, enable incomplete neutralization and basically escape of the virus. However, if you have efficient phagocytosis of the immune complex by phagocyte, you have a destruction, and then it's really game over for the virus. And that has been shown actually by Ken McCulloch, who I was working for many years at our institute, that in a mouse model, if you remove the phagocytes from the mouse model, even injection of neutralizing antibodies are not protective anymore. And he also showed in those papers that also non-neutralizing antibodies can mediate protection in a mouse model at least. So already in those days, there was an indication for a role of non-neutralizing antibodies. Now let's get to the thing that is important, was important to us at the moment. I still think it's important that we have to find good methods for vaccine matching, because that is crucial for vaccination against FMDV. So in the past, there was a lot of in vivo testing done, which I have here actually in the second point, which you do in cattle. There are guidelines for that, and actually, this is of course ethically problematic. It's very expensive, and what we know is also, of course, you have poor statistical power because you cannot have a lot of animals. So the gold standard, I think, is really in vitro testing, where you vaccinate the cattle and you test the serum from that cattle in a virus neutralization assay, and you determine the R1 value, which is the ratio between the reaction to the homologous strain and the heterologous field strain. And you want this to be as high as possible. And so this works well, but the problem is, of course, that it's not absolute. And so you can have quite low R1 value and still good vaccines. And so looking for other possibilities was the aim when we initiated that project. And we were particularly interested in the function of opsonizing antibodies. So what do they do? What do we know about that in FMD? As I said, they redirect immune complexes to phagocytes for final elimination of the virus. That has been shown in FMD. They change the tropism of FMDV towards macrophages and dendritic cells. There are studies by us and also the group of Brian Charleston in cattle that show that. That results in a abortive infection and destruction of the immune complex, but also activates the cells for enhanced antigen presentation to T cells. And it activates plasmacytoid dendritic cells, which is a specialized cell type producing enormous quantities of interferon alpha. And so you get those high interferon alpha responses by FMDV that is otherwise not able to induce interferon alpha. And that could be well happening in vivo and could be a mechanism of the function of antibodies indirectly. And the last point is something that we also published 10 years ago, was that those opsonizing antibody are cross-reactive, and you will see that they're even cross-reactive against a broad several serotypes of FMDV. So this is the paper I just mentioned, and you can see that basically we took plasmacytoid dendritic cells and we stimulated them with virus, FMDV virus that was either complex with immune serum or naive serum. And you can see the dilution curve of the serum in log 10 folds, and you see the kind of response we get. So you see that there is opsonization that we read out. The readout is interferon alpha, and that was a serum generated against a no serotype, and you can see there's also reactivity where you get interferon alpha responses against an A serotype of FMDV. So the summary of what we tested at that time is shown here. So you see there were a couple of A serotypes where we got opsonization. So that is in the right column. You see here the titers, log10 titers. So you got opsonization in absence of neutralization against those A serotypes and also ASIA1, but not C1 in that instance. Now, the question that we addressed for the data I want to present today is, what is the relationship between neutralization and opsonization at the monoclonal level? Because in those old studies, we used polyclonal sera. And do we also see that cross-reactivity amongst different serotype at the monoclonal level? And of course, relevant to the main question is, what is the potential role of those opsonization antibody in protection, and does the measurement of those antibodies inform us on vaccine selection in any way? So the system that we used is relatively simple. We took a well-established, well-known murine macrophage cell line, the raw cells, and for the cattle sera, we engineered them to express bovine CD32, that's Fc gamma receptor R2, so a C receptor for IgG. And what happens basically is those cells are resistant to FMDV, but if you complex with a monoclonal antibody or cattle sera, if you take the CD32, bovine CD32 expressing cells, then they get infected. It's an abortive infection that results in a cytopathogenic effect, and we measure that by flow cytometry. So first of all, the study on the monoclonal antibodies. So these are the monoclonal antibodies we have used. They're all from Emiliana's lab in Brescia in Italy. They're quite old antibodies. Some of you might know them. You see they're against O1, the old O1 Switzerland, O Manisa, O Malaysia, Cruzeiro, and so on. And some of them are neutralizing and some not. And for some of them, we know the epitope and also the isotype. Now, this is the first data set that I want to share with you on the anti-O antibodies, and it's showing you the opsonization activity of those antibodies. So on the left side, you see antibodies that were classified as neutralizing in terms of being able to neutralize the homologous strain against which they were made. So you can group those antibodies in non-neutralizing antibodies if you take heterologous virus, so this is the upper row, and neutralizing antibodies, so cross-reactive antibodies in terms of the neutralization assay. So for example, if you take the antibody D9, it's reacting with all those O serotypes at high level, and it's also opsonizing. So when you're up here, it means that 10 microgram per milli, it's opsonizing at up to 10 microgram per milliliter. And so this is, the red color shows you the ELISA reactivity. So red means high, basically 100% reactivity as with the homologous virus in a trapping ELISA. And if they're blue, they're basically negative in the ELISA. And so basically what you see here is that the only monoclonal antibody that did not opsonize was an IgM, as expected, because we are talking about Fc gamma receptors here, so that fitted well to what we are expecting. And then you see some antibodies that are up here, the 1C6, the 1C6, and then the 1C12, that were non-neutralizing against the viruses that you have on the x-axis, but still opsonizing, already indicating that you have a broader reactivity. Then on the right side, you see basically antibodies that were non-neutralizing against homologous strain, probably against internal proteins of FMDV. They are, of course, not opsonizing because they simply cannot bind to the virion. So for the A serotype antibodies against A serotype viruses, the picture is similar. Now we have a little bit more antibodies that are opsonizing, you see up here, but they are but not neutralizing against that pair, antibody-virus pair. So again, confirming that you have a broader reactivity, you can get reaction in the opsonization assay, but you're negative in the neutralization assay. Again, if you take an IgM, such as the 5C3, you don't have any opsonization. And if you take non-neutralizing antibody, you also have no opsonization. Now, what happens if you have a closer look at the epitope that is recognized? To do that, we had a look at the D9 antibody because it's a very old antibody and the epitope is very well known. It's recognizing the linear epitope on the VP1, the site 1 epitope. So that's the receptor binding site with the RGD sequence. And you can see that that antibody was highly opsonizing against all O serotypes, but also highly opsonizing a lot of those A serotypes that you see listed here. It was weak against A24, did not react with ACR1 in terms of opsonization, but did opsonize the C1 strain that we had here. So very broadly reactive in that assay. So what are the epitopes that are potentially recognized by these tests? So this is basically the epitope that is where the D9 is binding. You have the RGD sequence. You see in green the kind of amino acids that need to be conserved for binding with D9 in green and red, of course. And what you can see is the RGD. Then you see a leucine here, another leucine here, and the lysine here at this position. And if that is conserved, you have, of course, very high levels of neutralization, but also opsonization. Now, if you look at the viruses that are then below, where you have the A serotypes, you can see that basically the lysine here is not there anymore. You don't have neutralization anymore, but you still have opsonization. So this RGD, probably with those leucines here, is enough for the opsonization. And if you then start to lose sorry, that was too fast. If you start to lose the leucine here at this position after the RGD, then you also start to lose the opsonization activity, clearly demonstrating the relationship between affinity, that is the low affinity that is required for the opsonization assay compared to the neutralization assay. So now how does that translate into a vaccination and a correlate of vaccine-induced protection? So to look at that, we use sera from 2 vaccination experiments, vaccination challenge experiments that we did at our institutes, and these were actually the last one we did. And they were a consequence of the last FMD outbreak we had in Europe that was in Bulgaria in 2011, so 11 years ago. And we simply tested our vaccine that we had in our vaccine bank because we wanted to know, in case the virus spills over to Switzerland, is there any sense in vaccinating? So that was a classical vaccination trial, a one-shot vaccination trial following the OIE guidelines and challenge with the old Bulgaria virus. And so you can see here on that slide, if you compare the red and the blue, so red is not protected, blue is protected. Each of these dots are individual animals. You can see there's a lot of red. So basically about half of those animals were not protected. And it's also a titration of the vaccine dose. So here from left to right, you have full dose, quarter dose, and 1 in 20. So 1 in 20 does not work at all. And the full dose, there were 3 out of 4 animals protected. Now, what we're interested here is what is the relationship between the dose titration of the vaccine in vivo and the readouts. So here you see the neutralization titer. You can see from the p-value and the R-squared value, there is no correlation. However, if you look at the VOT, so the virus neutralizing titers of that sera, in the assay I presented you, you see there is correlation. So this test correlates to a vaccine dose and is suitable to assess maybe the quality of a vaccine in terms of its antigen payload, but maybe also in terms of its the quality parameters of the vaccine. Now, the second vaccination trial that I would like to share with you was basically a follow-up of that, where we then took a new vaccine provided by Merial at the time, and that was a vaccine that contained 2 different antigens, the O3039 and the O1 Manisa, or a combination of the two and just one dose, full dose vaccination. And you can see in that all animals except one, again in red, were protected, and that was in the 030-39 group. You can also see that the neutralizing antibody titers were low in that group, indicating that this is a bad vaccine match. If you look at the virus opsonizing titer, you cannot see that well that this is a bad vaccine match, and this is quite logical because those antibodies are broadly cross-reactive, so they're not suitable to distinguish between good and bad vaccine match. So the good match would, of course, be the 01 Manisa, where you have higher neutralization and where you had all animals protected. So we pooled all those data, and so the data from both experiments, and asked the question, is there a correlation between the virus opsonizing and the virus neutralizing antibody levels? And the answer is not. Probably if you would do that by just having one type of vaccine, you would have a correlation, but we had different antigens in there, so you lose that correlation because the reactivity is so different between the two tests. And then the follow-up question is, well, is there a difference in terms of predicting protection in the two tests? And indeed, both tests can somewhat predict protection, at least the values for the neutralization assay are not surprisingly higher as for the protected compared to the non-protected animals, that's what you see here on the left side. If you look at the VOT, this was also the case, but the p-value was not as good. So I would like to stop here and summarize what we have, what we know so far. So we can conclude that all antibodies that are able to neutralize the homologous virus were also able to opsonize, but non-neutralizing antibodies cannot. And of course, you need to have an IgG antibody to opsonize. Opsonization is broad and can even go across serotypes, and that's at the monoclonal antibody level. And if you have high affinity antibodies like the D1, you see that can be quite amazing how well that works. And so I think our data shows that the minimum epitope for opsonizing antibodies is clearly less stringent than for neutralization. And that is understandable if you think at the reaction, at what they need to do. So they don't need to prevent the virus binding to the cell, to its cellular receptor. They simply need to capture the virus, then to bring it back to the phagocytes for phagocytosis. And there's no interaction or no competition between the virus binding site and then the Fc part that binds to the Fc receptor, because they are opposite and there's no steric hindrance possible. So, and the final point, I think, is that I think measuring opsonizing antibody levels is interesting in terms of understanding vaccine potency and quality, but I don't think it's a good idea to do that to select vaccine in terms of selecting the proper vaccine antigen. But I also must say that, of course, there are more studies required to make final conclusions, because you have seen that this was only a limited number of sera. And I think also if one wants to use that test, one has to do more work on using an assay and establishing an assay where you have an easier readout, for instance, in a microtiter plate. So I would like to stop here and acknowledge all the people that have done the work, so that they are listed here on that slide here. Most of the work was done by Heidi Gerb, a technician that has retired, and some students that were at that time in my lab. That was Reka Schmidt and Nils Lanes, and also got support from Satya Parida with lots of discussion. And he also sent me some sera, but I was not able to include them into that study. And I would like to thank the people from Brescia for a long-standing collaboration, in particular, Emiliana Brocchi and Santiana Grazioli, and you for your attention.
Thank you so much, Arthur, for this very interesting presentation. We are taking questions from the Q&A, but as I am presenting and moderating, I can start. I find it, really interesting what you say that, well, neutralizing antibodies are opsonizing antibodies as well. And when you don't have neutralizing antibodies, sometimes you can achieve protection by opsonizing antibodies. And we also seen that there is some kind of relationship between the isotype and the neutralization titers or the avidity titers, and that doesn't make much sense if you think of the FAB alone. But I wonder if you have, if you know the isotype of the antibodies that you have been using, or if you have any special idea if a higher IgG1 titer, for example, is more related to opsonizing titers than other isotypes.
No, I cannot conclude on that. The problem was that those monoclonal antibodies, they are all against different viruses. So if they are lower or higher in their opsonizing activity, I don't know if it's coming from the isotype or because the binding affinity is just different. So it's difficult to conclude on that. But I think it's a very important question because we know that certain isotypes bind better. Then there are also fucosylation, so glycosylation of the antibody molecule that has an impact on binding to Fc receptors. So that could be also important as if you want correlates of good opsonizing antibody. But then we have to probably also do that in the species of interest, so either cattle or pig or the species of interest, and to find out which are really binding better in cattle to cattle Fc receptor. I don't know if that worked. Somebody tried to do that work in cattle.
Yeah, no, that's something I think we need to do. I take one question from the audience and then I pass it over to Mariano for the next. So the first question from the audience is from James Su. He's asking if you have an explanation for why neutralization needs high affinity and high concentrations of antibodies.
My explanation is that the foot and mouth disease has a very high affinity to its receptor. So there's if the binding to the receptor on the cell, the integrin receptor is very strong, then you need very high affinity to stop that. And so you need either a high affinity or a high concentration of antibodies. And with the Fc receptor or the opsonization, you just need to bind, if the antibody is somewhere, far away from its cells, you just need to capture it. And to capture it, you don't need a lot of antibodies, but to prevent it from binding to its or low affinity antibodies, but to prevent it from binding to its receptor, there you need antibodies that are at least higher than the virus affinity to its receptor.
Yeah, that's right. Yeah, and that's what we see in the practice as well. So when you try to measure protective antibodies against non-protective, that's what we see. So I pass it over to Mariano, so you can make a question and take some from the audience as well.
Thank you very much, Artur. It's really, really nice to have your presentations among our seminar series. So we are very happy with that. My question is, what is your opinion on the hypothesis of deceptive imprinting? That is it possible to, in the case of FMDB, to redirect the immune response by erasing the immunodominant epitopes, and that is it possible to broaden the antigenic recognition of these responses, or it's just another theory?
I think it, in theory, it should be possible. I mean, I think there's a paper that came out on HIV where they did that, with those conserved epitopes that are probably, somewhere hidden, not very accessible. So they're poorly immunogenic. And that's why you don't get broadly reactive antibodies. And so what you need to find is a way first to identify those epitopes and then by repeating vaccination. So basically, they did the monkey studies where they were vaccinating, I think every second or third day over a long period of time. And finally, over several rounds of affinity maturation in the lymph node, they got them high enough. And I think something like this is possible and probably requires, several rounds of immunization. But I think it should be possible to achieve that, certainly not with a 1 or 2 shot vaccine.
Great. Thank you very much. Alejandra, do you want to get some of the questions from the audience?
Yes, we can take another one from the audience. This is from Sabrina here from Buenos Aires. And well, she agrees with us that it was a very interesting presentation. And she's asking if could we say that the broad opsonizing activity in relation to the similar epitopes in what you have shown in BP1 could explain that using vaccines with the strain of different serotypes may help to cover a wider spectrum of field strains? She wanted to know, what do you think about this?
Well, actually, I asked myself the same question, because I don't, we had our last outbreak here in 1965 or something like this, and we have never vaccinated. So I didn't see any data. Probably you guys in South America or Asia are better placed to answer that question. I mean, the question is, do you see a partial protection across serotypes? I don't know if that is the case. I was assuming not, and I was assuming if you just have that reactivity, it's not going to be sufficient to measure significant protection. But maybe I'm wrong. I don't know. Is there evidence for having a partial protection from if you vaccinate with a different serotype?
Yes. Well, what we have is we have published with Mariano a paper short time ago showing that when you have a trivalent vaccine, you get broader protection against a different strain that is not included in the vaccine. Yeah. But that's just an experimental evidence from a limited study. But we saw that's why I think Sabrina is also asking, when you broaden the amount of epitopes available to the immune system, sometimes you get more cross-protective antibodies.
I think I would agree to that. I mean, on top of that, of course, you have T cells, and you increase the epitopes that are possible for T cells. The more broader you do that, the better you have a chance of covering different haplotypes in cattle. And so there are many explanations why that could work better than a monovalent vaccine.
Yes, I agree. Do you want to take another question, Mariano, from the audience?
Let me see. Well, Karen De Luca is asking, is wondering, do you expect to follow up that work, mostly in swine, where the correlation might be even less absolute in what is called the gray zone?
Yeah, it would be nice to follow up the work. At the moment, you see all the funding goes to African swine fever in Europe, and I think it will come back, and I think it would be nice to follow that up in other species as well, and get more data on those functions.
Yes, it would be nice to follow up. We completely agree, and that's the GFRA also to help, perhaps. We have another question from the audience, and it says, as I know, all IgG can bind to macrophage. If the antibodies can bind with FMD surface, it would be able to opsonize macrophages. Why some vaccines that don't match with the field virus but induce IgG could not protect the animals?
Well, I think you need those neutralizing antibodies, and there might be low quantities of neutralizing antibodies that you don't know that you miss maybe in your SNT test, and you think they don't, but they are still there, or the B cells are on the way to make them. or you might have memory cells that then quickly pop up and make them. And I think they're still essential in protection. I don't believe that you can protect cattle just with T cells and just with opsonizing antibody. So I think they're an essential correlative protection to measure neutralizing antibodies, and I think you really need them. Otherwise, you will not get protection against a virus like FMDV.
Yeah, and there are also perhaps differences between species, as it was said before. So perhaps...
Yeah, so, I mean, of course, those old mice studies from Ken, that was mice, so we cannot extrapolate from that on other species. Yeah. So I would still say really, neutralizing antibodies are the most important correlative protection, clearly.
Yeah, but I think as well that mapping all those epitopes that are cross-reactive and that are inducing cross-protection is paramount, I think, for us in the FMD world. So I think this is a nice approach to see also the way they work and how they interact with protective and non-protective antibodies. So I think it's a very, very nice approach. I don't know, Mariano, if you have anything else. There's nothing else from the audience so far. But if you want to...
I have just a last question. Arthur, have you found any relation between the opsonizing ability of the antibody and the affinity towards the epitope?
No.
No.
You can have high affinity antibodies and they still opsonize perfectly. Okay. So it's I think up to a point, of course, you need sufficient bindings so that you're on the side of having more virus that is complex compared to free virus. But then you would probably reach a point where it doesn't matter if you further increase the affinity. Then probably you come to a point where it's more important what does the FC part look like, which isotype, how is it glycosylated? But I don't have data on that. That is difficult to assess in the lab. That requires a lot of work. But that would be my guess.
Yeah.
I mean, the data looks a bit like that, that you just need a little bit of binding and it's sufficient to get very good opsonization.
Yeah, I agree. And yes, and I'm still surprised about why IgG1, for example, is more related to neutralizing titers than the other isotypes, and that I don't know how that makes sense so far.
I think we really need to do research in cattle.
Yeah, there's a lot. There's a gap there.
Yeah.
Yeah. So, Mariano, is there anything else?
No, not from my side.
I think here is Aldo Decker as well saying something, so I will read it out. Aldo says, we infected 2 cows 3 times with different serotypes. Every time we observed full-blown FMD. But at the end, the serum also neutralized the serotype which was not used for infection. So cross-protection between serotypes is not easy, although broader within serotype neutralization has not been tested. Would it be interesting to test the sera, this sera in your assay?
Yeah, I think it could be interesting, but then I would need to really would need to have those different strains and test the opsonization activity against the different strain and against the different, challenge virus. It was always one challenge virus.
Yeah.
Yeah, Aldo, you can contact me and send me, the experimental outline.
Yeah, I think he put the experiment outline or all he has available here in the Q&A box. I don't know if you can see it, Arthur, but I can copy and paste it for you.
Okay, I can have a look at that.
Okay. I think if there's not anything else from the audience, I don't know, Mariano, if you have something else to say.
No, not from my side.
Apart from thanking Arthur for nothing else. Okay, so
we did have
once again.
one more question pop in, Alejandra.
Yes.
Oh, there is one more.
One more question, yeah.
There's one more, okay. So I lost that one from Charles, sorry, Charles. So Charles Poem from Canada, he's asking, high-potency vaccines with lower values can still protect against strains of the same serotype. Could opsonization be playing a key role here?
Yes, exactly. I would say yes. That's at least, you know, from the immunological point of view, yes. But the difficulty is, of course, always if you have that situation, it could be opsonizing antibody, it could be T cells. It's extremely, or probably it's going to be a combination of several things. That's how the immune system works. But I would say,
yeah. Sometimes we just try to make it simple, but it's not simple at all. So measuring one characteristic of the immune response doesn't give an answer. Sometimes you have to work with many assays to have an answer for that. Yeah. So nothing else. So I lost that one. I was lost in all the long list that Aldo posted, and I didn't see Charles' question. So I think with that one, we are done already. So Arthur, thank you very much for joining us. Hope you join us again in the future. Hope you become a member as well of our GFRA.
Yes, we should. I tried once and it got lost somewhere. I will do so. I will push out. We have a new director. I will push her to take care of that.
Okay.
Yes, that would be great to have you on board. And thanks everyone for being here. This is the last one of the year, so we meet again next year. But don't go, because I think that Tessa has something else to say. So Arthur, thank you very much. We're very happy.
Thank you,
Alejandra and Mariano, for the invitation and sharing the meeting.
That was great.
Everybody for joining.
Thank you so much.
Thank you all. Yes, there will be a feedback survey that will pop up when you close the meeting. So please do fill that out so that we can continue to grow and improve the research webinar series in the future. Thank you again to Dr. Summerfield and our moderators and GFRA, and have a great rest of your day.